नमस्ते दोस्तों, मैं हूं चैतन चक्रधारी पर आपका स्वागत है हमारे इस शैक्षिक चैनल में। यह चैनल विशेष रूप से उन छात्रों के लिए बनाया गया है जो CSIR UGC नेट लाइफ साइंस, गेट, IIT जैम, CUET, BSc MSc एंट्रेंस तथा अन्य प्रतियोगी परीक्षाओं की तैयारी कर रहे हैं। अगर आप लाइफ साइंस में करियर बनाना चाहते हैं तो यह चैनल आपके लिए सबसे उपयुक्त जगह है। सब्सक्राइब करें और अपनी सफलता की शुरुआत आज ही करें। 1. The enzyme responsible for unwinding DNA at the replication fork is A. DNA polymerase 1, B. DNA gyrase, C. DNA helicase, D. ligase. Answer C. Two, Okazaki fragments are formed on the A. leading strand, B. lagging strand, C. both strands, D. RNA primer strand. Answer B. Which of the following enzymes removes RNA primers during DNA replication in E. coli? A. DNA ligase, B. DNA polymerase 1, C. primase, D. topoisomerase. Answer B. Four. The main replicative DNA polymerase in E. coli is A. DNA polymerase 1, B. DNA polymerase 2, C. DNA polymerase 3, D. DNA polymerase 4. Answer C. D. 5. Telomerase adds repetitive sequences to the ends of eukaryotic chromosomes using A. DNA-dependent DNA polymerase activity, B. RNA-dependent DNA polymerase activity, C. DNA-dependent RNA polymerase activity, D. RNA-dependent RNA polymerase activity. Answer B. 6. The sliding clamp protein in prokaryotic DNA replication is A. SSB protein, B. beta clamp, C. pCNA, D. DNA. Answer B. Seven. In eukaryotes, the functional homologue of the bacterial beta clamp is A. pCNA, B. DNAB, C. MCM, D. ORC. A. Which DNA repair mechanism removes thymine dimers caused by UV light? A. Base excision repair, B. Nucleotide excision repair, C. Mismatch repair, D. Recombination repair. Answer B. The SOS repair system in E. coli is induced by A. DNA glycosylase, B. Rec-A protein, C. Photolyase, D. ligase. Answer B. The direct reversal of UV-induced thymine dimer is carried out by A. DNA polymerase 3, B. Photolyase, C. DNA ligase, topoisomerase. Answer B. Mismatch repair in E. coli distinguishes parental and daughter strands based on A. Histone modification. B. DNA methylation. C. RNA primers. D. DNA ligase activity. Answer B. (12) Which enzyme seals the nicks in the sugar phosphate backbone during DNA replication and repair? A. DNA gyrase. B. DNA ligase. C. Primase. D. Helicase. Answer B. (13) The holiday junction is an intermediate structure in A. DNA replication. B. Homologous recombination. C. Nucleotide excision repair. D. Mismatch repair. Answer D. (14) In E. coli, the protein that initiates homologous recombination is A. Rec A, B. Lex A, C. Uver D, D. NAK. Answer A. Double-strand break repair in eukaryotes often involves A. Base excision repair, B. Non-homologous end joining NHEJ and homologous recombination, C. Photoreactivation, D. RNA editing. Answer B. (16) The enzyme DNA topoisomerase 1 relieves supercoiling by A. Cutting both DNA strands, B. Cutting a single strand and rejoining it, C. Adding negative supercoils, D. Using ATP hydrolysis. Answer B. (17) Proofreading activity of DNA polymerase is carried out by A. 3' exonuclease activity, B. 5' 3' exonuclease activity, C. 3' polymerase activity, D. Ligase activity. Answer A. (18) Which of the following is an error-prone DNA repair system? A. Photoreactivation. B. Mismatch repair. C. SOS repair. D. Nucleotide excision repair. Answer C. (19) The origin of replication in E. coli is known as A. ARS, B. ORIC, C. ORC, D. loop. Answer B. (20) During homologous recombination, branch migration is facilitated by A. DNA helicase and Ruve B complex, B. Ligase only, C. Photolyase, D. Telomerase. Answer A.